Hello and welcome to this new playlist, Molecular Dynamics. So in this playlist, I will discuss the theory and visualization, data analysis, and how to efficiently use software related to molecular dynamics so that you can use it in your research projects. So in this first lecture, I will discuss the scope of this little course, and that will introduce you to the theory and practical aspects of molecular dynamics. So many times we get preoccupied with how we will use the tools associated with a research methodology. For example, when someone start to learn molecular dynamics, first question they ask that how do I run molecular dynamics? But before running into that, we should ask ourselves why we will do molecular dynamics, or what can I do with molecular dynamics? Is it relevant? Is it a relevant tool or methodology for my research project specifically? So about that, if your project is about polymers or biomolecules and you want to simulate mechanical properties or behaviors or biochemical phenomena at nanoscale, and you want to capture nanosecond duration of phenomena, and if your system contains a few hundred atoms to a few million atoms, and if you want to address some biophysical or biochemical or mechanical phenomena, then possibly molecular dynamics will be in your scope. Many a times molecular dynamics is used when the experimental method is not viable because of the inconvenient structures or very extremely small scale. And sometimes also possibly you cannot model it computationally with continuum tools like finite element methods. So specifically to capture biophysical, biochemical, or mechanical phenomena at nanoscale, at the very atomistic level, you want to use molecular dynamics. So in order to get you understand what it means, I'm from my PhD research was concentrated on brain injury. So let's pose you a hypothetical research question. Let's say you want to solve a problem related to brain injury. And a simple hypothesis is that you want to cure a specific type of brain disease, and you want to solve that research problem by using computational mechanics or computational tools. So in layman terms, if I want to pose that research question, let's say a person is injured on the head. At macro scale, you see some injury, but what happens at the nanoscale? There are so many interactions that propagate. Some are mechanical in nature. Let's say subaxonal components that get injured, and also very biochemical interactions or reactions that propagate after an injury as a response to the injury. So let's say you can, let's say you want to capture the structural changes, you want to quantify the injury or threshold the injury, and you want to simulate that kind of atomistic level of deformation of subaxonal components. And you want to know what to do at this damage or injury scenario if you want, if you can stop the damage propagation. So this is our hypothesis. Just to give you a little background, when a brain disease or injury occurs, there are several changes that take place in brain. For example, in cases of Alzheimer, there is a loss of brain cells, and there are other damages that take place both in macro scale and nanoscale. So these are some physical phenomena, but there are also some biochemical reactions associated with it. So let's come to a very specific example. In case of brain disease, there are amyloid beta pathology, that is amyloid beta fibrillar aggregation that occurs. And amyloid beta is kind of a protein, just in simple terms, that deposits at different parts of brain as a response to the disease in beta sheet conformation, as we can see here. And let's say as a researcher, you are proposing that if you can make amyloid beta to interact with some protein X, then this beta sheet actually breaks, and then the injured brain or diseased brain come to the normal state. So in order to beta sheet inhibition, you want to see how this kind of interaction occur between the amyloid beta sheet and the protein, the X protein, the unknown protein. It can, whatever it is, probably some normal protein present in the central nervous system that takes part in so many biochemical phenomena. So in order to simulate that kind of interaction between the amyloid beta protein and the X protein, first what you want to do is molecular docking. You want to use this amyloid beta as recipient and that X protein as ligand. Then you perform their docking. For example, how do they interact? How do they come close together? And you want to know their energetically favorable poses by submitting their structure in docking servers. And what the docking servers do, they actually populate several combined structures. There are several popular molecular docking servers like PatchDoc, FireDoc, or ClassPro, and there are other tons of online servers to perform molecular docking. So in these molecular docking packages, they could be software or they could be servers, online servers. Popular software could be Autodoc Vina or Rosetta Doc, and the online servers could be PatchDoc, FireDoc, or ClassPro, GramX, Haddock, something like that. So what are these packages doing? They are actually creating a combined pose between multiple proteins. In our case, the amyloid beta and the unknown protein X, and they're creating a combined pose that we call docked pose based on some specific principle. Every server or software, docking software, they work on different principles based on published methodologies. Some common principles are shape complementary principles like lock and key. In case of two proteins docking between two proteins, one protein can work as a key, one protein can work as a lock, and at these ridges and rises based on the 3D construction of the protein, another protein will interact accordingly. So this is called shape complementary principle. And in some software, they use rigid body principle, like they keep one protein fixed and then they create a 3D grid around the protein, and the other protein is put on different coordinates on the grid to see which pose is energetically favorable. And some also use other thermodynamic or biochemical parameters like binding energy. And the output is you get 10, 100, or thousands of docked poses with confidence scores, and you start with the structures with the good confidence score. And possibly you can then you come to the square one of molecular dynamics. You can use those docked poses as the preliminary coordinates to run the molecular dynamic simulation to simulate these combined poses behavior. So in molecular docking, you're getting the initial poses, and in the molecular dynamics, you are simulating their interaction to see whether it's stable or not. You can run for several nanoseconds. And what you want to observe, let's say you have simulated let's say you have got the initial poses between two proteins, amyloid beta and G protein. Previously, we have called X protein. Now, let's say you have got a specific protein, G protein. So let's say after simulating them for several nanoseconds, what you want to observe is that you want to observe the system stability, their interaction, other changes like thermodynamic changes or something like that. And how do you know that your hypothesis is correct? let's say you have simulated two systems. One is with amyloid beta only, and you have simulated it for 100 nanosecond. And from the RMSD curve, you see that amyloid beta itself is actually very unstable because it has actually deviated too much from its reference coordinate. And when the G protein, our X protein, is present in the system, it becomes very stable. So what you can see that instead of making the amyloid beta sheet unstable, it has made it more stable. So your primary hypothesis is wrong. And you have also followed the other established principles of research, like that you have repeatedly run this simulation several times, and you have maintained, and you have run it for sufficiently long time, and you have applied other reliable MD parameters, molecular dynamics simulation parameters like force field. Then you can say that your primary hypothesis is wrong, and this particular G protein is not contributing to curing that disease by disintegrating the amyloid beta. So this is kind of a specific hypothetical problem that you can solve in terms of biomolecular system by using molecular docking and dynamics. And I have mentioned that there are several packages of molecular dynamics. The popular ones are ESR, LAMMPS, NAMD, and GROMACS. The last three ones are free to use. These are open source. ESR is a paid software, but their overall, their working principle is kind of the same. And in those molecular dynamics simulation packages, there are sometimes there is a graphical interface, sometimes there is not, sometimes you have to work in command line environment, and there are sets of force fields. What are these force fields? I'm describing it briefly in the next slide. The molecular dynamics simulation packages, let's say what you are working with. Sometimes we generally ask, like, what are inside those molecular dynamics simulation packages. So here I'm showing three examples of MD simulation packages. One is ESR, one is LAMMPS, and one is NAMD, for which you can use VMD as a graphical user interface. Here are actually three files that you can use to run molecular dynamics simulation. One is the structure file of the protein that has that contains all the coordinates of the atoms. In case of LAMMPS, that is actually data file, and in ESRs you can actually use a macro to in order to load all the information. In NAMD, you also use a configuration file where you define all the things you want to do on these PDB file, the structure file, and as an output you get a trajectory file. In case of LAMMPS, this configuration file is called input file, where the syntax is kind of different. As per every molecular dynamics simulation package, there is different syntax that you have to follow. And in the trajectory file, you will have all the things that you wanted to calculate, for example, the velocity or deformation of the structure. And in case of LAMMPS, you'll get a dump file where you have the same things as output. So in these molecular dynamics simulation packages, you are actually giving the system the structure information, and in the input file or configuration file, you are setting up simulation box size and the simulation environment, what you want to do, the time step, the duration, all kind of parameters. And as an output, you are getting the trajectory containing all the updated parameters or velocities or thermodynamic parameters that you wanted to calculate, that sort of things. So that's how, in general, in common terms, the molecular dynamics simulation packages work. And in those packages, there are sets of force fields. Force fields are actually functional form of energy, how you want to define the system in terms of energy. The popular examples of force fields are charm amber, Gromos and Martini, charm, amber. These are kind of more parameterized for protein systems. And also there are Tersoff or hybrid potentials that could be more suited for material science projects. And in these functional forms of energy, what you are doing is that you are defining parameters like the structure. In a structure, think about a protein structure. There are atoms, and between those atoms there are bonds, there are angles, there are out of plane interactions, and there are also non-bonded interactions there for their specific position in the 3D space and for the specific charge. So in a combinatory way, you can actually express their functional form of energy, where you can describe their bonds, their angles, their dihedrals, and all the non-bonded interactions. Sometimes they come empirically, sometimes they can be developed by machine learning algorithms. And sometimes you make some tweaks and turns according to your need for your specific protein system or material system. So in order to solve a biomechanical or chemomechanical problem, as we wanted to solve, we want to address several questions. If we go back to the first question that we started with, whether the injury affect the amyloid beta and our protein G, this kind of interaction, whether it gets affected with the injury, and whether we can quantify this deformation by using computational mechanics principle. In order to calculate the deformation and stress and strain, we use computational mechanics inside the molecular dynamics simulation tool. And we also want to make sure whether our length scale is atomistic or continuum. If our length is too large, probably molecular dynamics simulation is not suitable for us. Probably we have to model it in continuum scale. And as we have mentioned that in molecular dynamics simulation, we want to observe the stability. If we want to determine mechanical property, we want to show force displacement curve or stress strain. So basically we're stretching the protein and getting the stress strain curve. Probably we want to show the strain rate dependence. So we are stretching the protein with different strain rate, and we want to see whether one protein gets separated from the other. So we get the separation criteria. So these kind of biomechanical or chemomechanical parameters we can get from molecular dynamics simulation. And as a result, we can get the damage threshold in subaxonal components. We can get some novel insight into damage mechanism. And we can also validate the force field. Sometimes we can see that one force field is performing better than the other force field for a particular research problem. So what we can see that from the injury, we can when a head is injured, there is amyloid beta sheet formed, and we can see that amyloid beta glutamate interaction is happening. And as a result, for this G protein, let's say hypothetically it's glutamate, and we can see that as a result, we have hypothesized that beta sheet will be inhibited and it will be cured. But we have seen that, let's say hypothetically, we have seen that there is nothing happen. The amyloid beta sheet are more stable when they're interacting with this G protein glutamate. So our principle, our preliminary hypothesis was wrong. So that's how we can practically address a research problem by using molecular dynamics. And the scope of molecular docking and dynamics is kind of broad because you can address several you can address a lot of research questions related to biomolecules and material science. And as a result, you can impact computational drug design and material development fields. So now that we have laid out the background, I will say a bit about the organization of the course. We'll start with molecular docking and molecular docking software and servers to see what kind of popular methods you can use to do a successful set of molecular docking through software and servers, and get a good theoretical background that you will need to understand what the molecular docking software and servers are doing under the hood. And before getting into the learning the software and tools on MD, we will discuss a little bit about the MD simulation and popular MD software and servers. And we will also show how to set up a molecular dynamic simulation system and how to visualize that system to see what's going on. And as a popular platform, molecular dynamic simulation platform will give an introduction to MD platform, LAMMPS. And we will also do some data analysis in LAMMPS, how to interpret the data that is being generated in LAMMPS, and we will get some hands-on tutorials to understand or get used to LAMMPS methodology. So that's kind of the scope of the course. I will see you in the next lecture. Hello and welcome to the second lecture of this course, Molecular Dynamics. Here I will give you a little brief introduction to molecular docking, because sometimes it needs to do molecular docking before you actually run a molecular dynamic simulation. So as you can feel that in the first lecture, I have introduced you to a concept of how to solve a research problem by using molecular dynamic simulation tools. And you may wonder that I don't have any background in this particular topic, but don't worry, we'll go step by step to know the theory and related software of molecular dynamic simulation. And many times what happens is that when someone is creating a lecture series on molecular docking on dynamics, sometimes students cannot relate to the topic because people are from different backgrounds. For example, sometimes material science people don't understand what I'm talking from biomolecular points of view, and vice versa. And sometimes people come from different major and different terminologies or jargons may not be familiar with you. Again, don't worry, we'll try to go through every topic and subject step by step, and we will segment it into little pieces so that you can understand, and we will learn all the underlying principles and cross-platform functionalities, and we'll take care of the jargons and terminologies you don't understand or you find difficulty to relate with. So if I'm talking about a biomolecular system and you're from a material science major, you will not have a difficulty to relate with it, because I will show you how to use that from your perspective. So as we have discussed that the organization of the course is, we will talk about the theory of MD and MD software and servers, but many times we have to run molecular docking before running a dynamics. So I will dedicate this particular lecture on molecular docking. So what is actually molecular docking? In docking, we determine the interaction between two biomolecules. For example, a target protein and a small ligand. And by molecular docking, we determine how they interact. What is their type of interaction, bonded or non-bonded perspective. Now, why do we do molecular docking? We want to get interaction inside. This is very relevant, particularly to biomolecular systems. And many times you want to do drug design and you want to know how a ligand particularly binds with the target protein or protease of a disease. So from those particular point of view, you want to make an initial system or with optimum coordinates. And for that, you do molecular docking. And how you can do this molecular docking? You can do it two ways, by use several softwares like Rosetta or Autodoc. And you can also use some online servers like PatchDoc plus FireDoc and Haddock and ClassPro. And they are all particularly parameterized for particular level of interactions. So you should first go through their documentation and publication page to know their underlying principle. And how do these molecular docking software or server work? Sometimes it works on a lock and key mechanism. For example, induced fit mechanism, like when a protein comes in the vicinity of another protein or biomolecule or a small ligand, the lock and key system, in a lock and key system, they actually lock with each other according to their 3D coordinate or their conformation in the space. And sometimes energy-related parameters or biomolecular parameters come into play, and sometimes you work with knowledge-based structures. Again, these are parameterized for particular software or server. So this is an example. For example, when you work with PatchDoc, you submit the PDB file for a receptor molecule and a ligand molecule, and then you submit it in their server, and they give you a bunch of predicted combinatory structure of the protein and the ligand, and then you refine it with FireDoc to get more energetically viable or structurally viable structures. And this is another interface of ClassPro, which is particularly parameterized for protein-protein docking. Here you also upload the PDB ID or the file for the receptor and ligand, and you apply some advanced options, if you will, and then you dock. There are also some other software like Haddock Rosetta, and every software has many related functionalities, like you can visualize or you can make really good tables of non-bonded interactions. This actually depends on how you want to proceed with your molecular docking. Molecular docking is particularly a different subject, and you can study independently from molecular dynamics, but many times, especially for biomolecular simulations, you want to create an optimized system before you run your molecular dynamics. That's why I have given you a brief introduction of molecular docking, and I hope that it will come in handy. And in next lecture will be the real beginning of our course, and we will start with molecular dynamics theory, and we will dive deep into what really happens in molecular dynamics when you create a system, what is actually going on. So let's go to the next lecture, and I will see you there. Thanks. Hello and welcome to the third lecture, what happens in molecular dynamics. As a flashback, we have already mentioned that we will give you a generalized introduction on molecular dynamics, and we'll go step by step on the underlying processes and cross-platform functionalities. So we are at the molecular dynamics series step of this course, and we will give you a short introduction on the underlying principles, what happens under the hood in molecular dynamics. And we'll also learn software and data analysis by molecular dynamics at the later part of the course. And we have given you a brief introduction on molecular docking servers and software in the previous lecture, as sometimes we need to do molecular docking before we actually run molecular dynamics. So why molecular dynamics? As you can see in the first lecture, I have given you a kind of a motivation in terms of a research question that we can solve several research questions, be it in material science or biomolecular system. And we can solve several problems, several length scale and time scale related problems. And also we can analyze interaction from molecular docking scenario, as we have mentioned in the second lecture. And we can observe those interaction, observe the stability of a system, and calculate several properties, for example, mechanical and force or displacement related properties, and so on. Sometimes we also dedicate molecular dynamic simulation to determine thermodynamic or electronic configuration related properties also. So
Why molecular dynamics? The answer is that we can do a range of tasks by using this tool. And if we talk about the scope, there are several simulation methods at our disposal. If we use density function theory, that actually deals with a very little time scale, for example, picosecond, and also very little length scale. So you can simulate nanometer length of system, and you can simulate it for several picoseconds, but you can deal with only a few number of atoms because you are working at the very electronic configuration level. But if you talk about molecular dynamics, you can see that the range in terms of time scale and length scale is quite diverse. You can simulate hundreds of or thousands of millions of atoms for nanometer and micrometer lengths for several nanoseconds to several microseconds, and you can determine atomistic to macromolecular properties. And if your system is too large, then you cannot be in this range of classical molecular dynamics. You have to do continuum level modeling, and you can determine properties of millisecond to hours. And generally, it is done for macro scale level of systems. A word of note is that sometimes you can also do, you can also make a bigger system by using a coarse-grained model in molecular dynamics. But in this course, we will stay in the classical molecular dynamics region, and we'll deal with only this level of time scale and length scale and number of atoms. That is, will be in the range of atomistic to macromolecular level system building. So what happens in molecular dynamics is that you actually solve Newton's equation of force. What you start with that, generally when you see a visualization of molecular dynamic simulation, you see a bunch of atoms vibrating in a simulation box. What is happening is that you start with an atomistic model, that is the initial structure, be it a material structure or a protein, and then you apply a force field that is actually an energetic state of the system. That is a bunch of rules in terms of energy that you apply to the system so that you can theoretically determine their trajectory. And you start with an initial position and velocity according to your system. And what happens is that you calculate your forces in terms of the energy of the system, the potential energy of the system. And then after applying this bunch of rules to the system, you update this, the atomic position and velocity of the system, and then you continue it. You do it repeatedly at a specific time interval. Then at the end, when your simulation ends, you get a bunch of trajectory data that you analyze, and from that you extract your output results, be it force, displacement, or stress or strain. In short, at a very high level, that is what happens in molecular dynamic simulation. So as you can see that if equal to ma is the governing equation in molecular dynamics, that is going on under the hood. You have an initial structure, you are applying a bunch of molecular dynamics theoretical conditions on the system, and you get the trajectory and motion of the atoms in the system. Why? You want to capture their atomic scale, behavior, and property. And when you combine those at a macro scale, you can calculate their macro scale behavior. So where does the energy come into play in this scenario? What happens is that you can actually define a system in terms of a potential energy function, and you can actually relate the force and the energy by this given relation, very simplistic relation where you can determine the force of a system from the energy of the system. And that's why when you are applying the energetic relation to the system, you are actually solving the F equal to ma equation. So you can get a sense that you have a structure and you're applying molecular dynamics conditions on that system to perform a simulation and get a set of trajectory data. So most important thing in molecular dynamics simulation is the energy function, that is the interatomic potentials. That is the bunch of governing rules that we're applying on the system. So where does the energy function come from? That's the topic of the next lecture. We'll go into detail of energy function, how we define a system's energy function in molecular dynamics simulation, and what are their types, and how are they generated, how do we get them. So that's what we will deal with in the next lecture. So I will see you there. Thanks.
Hello and welcome to the fourth lecture, Molecular Dynamics Force Fields, Pair Potentials. So we are continuing our discussion, the high-level introduction to the molecular dynamics theory to understand what really goes on in a MD simulation. And previously, we have given you a very high-level introduction and a very easy explanation of what happens under the hood in molecular dynamics, and we have explained that in a system, whether it contains a protein or a polymer or whatever it is, in order to define its structure and trajectory, you have to actually solve the fundamental F equal to ma equation. And by solving this force equation, you can actually define the system in terms of its potential energy, as the force and the potential energy are related through the equation that we have shown in the previous lecture. And the energy function that describes the state of the system is called force field, which is very important to define the system, particularly in molecular dynamics. And now we know that this kind of potential energy function, or in other words, interatomic potentials, is to be known when you want to define a system, or whether you want to run a molecular dynamics simulation. Now we will, if we go into a very detail of a functional form of a system, it will be very difficult to follow. So it's convenient or logical to start with the very simple one, the pair potential. So what is interatomic potentials? We already know that a functional form of the potential energy state of the system. That is an equation with which we're defining how the atoms are interacting with each other in a system. Let's say in a protein, there are so many residues that are interacting with each other. In this structure, there are some atoms and some bonds and some angles and dihedrals and non-bonded interactions. We want to define it by a functional form in terms of potential energy, and that is interatomic potentials. And as a combination, it's called a force field. And if it's a material science perspective, then you can think about a polymer in which the atoms are interacting through bonds and dihedrals and angles and non-bonded interactions too. Now, we want to know the interatomic potentials. We want to define the equation in terms of potential energy U. Where do we get this potential functional form? How do we know that our definition is correct? So sometimes it comes from experimental data. So when it comes from experimental data and based on which we can define our interatomic potential, it's called empirical potentials or empirical force fields. Some popular examples are LJ and Tersoff. Sometimes it comes from some experimental data, and some can be exactly calculated. In this case, it's called semi-empirical. For example, EAM or embedded atom potential. For fully empirical, some examples are LJ and Tersoff. For semi-empirical, for which some parameters you can calculate from experiments, some comes from your theoretical calculation.
you can also build a force field. An example is EAM. And sometimes you calculate exactly from the very electronic configuration perspective. And then it's called an ab initio, that is from the very fundamental level, from very electronic configuration level, you're calculating all the parameters in a force field. So how does a potential energy function looks like? It's a functional form, as we have already mentioned, and it really looks like an equation that contains the definition of the positions or bonds, angles, dihedrals, whatever it is needed to define a system fully. So in a very simplistic form, you can think about a potential, interatomic potential or force field functional form looks like something like this: AX plus BY, some variables and some constants. That's it. Now, when it comes to a system, it contains so many interactions, right? And considering those and calculating all the interactions and putting it into a functional form is very difficult to follow. So let's think about the simplest scenario here. In a simplest, from the simplest perspective, what can happen is interaction between an atom with another atom, that is between an atom pair. So when we define a simplistic functional form of potential energy in a system, it's called pair potentials, that is, which considers only two atoms, only interactions between two atoms. For example, let's think about this system, where if you talk about this atom and this atom, you are talking about their distance. Let's say this is atom I, this is atom J, and their distance is, let's say, when you count from this, you are saying it's Rij. So you will consider only interaction between these two atoms, and you think that all the other interactions are not coming into play. Although it's not true, but it helps us to simplify the functional form. And that's how the pair potential is defined like this, only considering the distance and position between two atoms. So possibly the most popular pair potential is Lennard-Jones potential, which in its functional form looks like this. It's a very popular functional form. And it actually defines the interaction between two atoms, that is, it defines pair potentials in terms of the attraction and in terms of repulsion. So there are only two terms in these functional form. And you can see that when the atoms come toward each other, they start to repel each other. And when they go far away from each other, they start to attract each other. That's kind of a very high level, to some extent, some non-technical introduction to Lennard-Jones potential definition. And it works well for noble gases, but it doesn't work for all the other polymers or alloys. So it's a very basic kind of pair potential, and you get a feeling how it looks like in terms of attraction and repulsion. So in short, a pair potential is defined by interaction with two atoms, and it's defined in terms of the attraction and repulsion that is going on between each other. So in the next lecture, in this lecture, we have actually discussed about an empirical pair potential. In the next lecture, we will look into a hybrid or semi-empirical interatomic potential, that is EAM. I'll see you there. Thanks. Hello and welcome to the fifth lecture, Molecular Dynamics Force Fields EAM. Here we'll discuss some semi-empirical pair potentials. And we are continuing our discussion on molecular dynamics theory, and previously we have introduced the simplistic interatomic potentials that considers only interaction between two atoms. But here we will make things a little bit more complicated, and we will dive into
the semi-empirical interatomic potentials, that is EAM. As we have mentioned that the functional form is called interatomic potentials or force fields that describes the energetical state of a system, and it can be expressed by a functional form. And the simplistic functional form that is pair potentials, or a popular example is Lennard-Jones potentials, that actually doesn't consider anything else but interaction between two atoms. And in Lennard-Jones potentials, it only considers the attraction and repulsion of the atoms when it comes to, let's say, atom I and atom J. And that's why it has only attraction and repulsion terms. And it only works well for noble gases. But if we talk about a semi-empirical embedded atom method, then it actually considers not only interaction between two atoms, but it also considers as if the atom is immersed in a field of electric charge. That's why it's called embedded, as if the atom is embedded in electron charge. And you can think about the electron configuration model for metals from your high school chemistry book, where it describes that as if the atoms are immersed in an ocean of electrons. That's why the whole system has a charge, and this model is particularly well set for metals. But you can see that there are several pros and cons in this system. It's yet simplistic. It doesn't consider all the interactions that is present in a system, but it considers a long-range Coulombic interaction because it's thinking that as if every atom is actually immersed in an electron density field. So that's why this kind of pair potentials or interatomic potentials actually works well for metals. So as we can remember that previously in the pair potential, there was only one term, right? The empirical term, but you can also calculate the electron density term. So that is why that is what is getting added to the pair potential. And when you consider this portion and add it to the experimental portion of the pair potential, then you get the EAM definition of potential. So you are not only considering the interaction between two atoms, but you are also thinking about that the atoms are actually immersed in an electron gas field and it has an electron density rho. And that's why you are considering another term to fully define your system. So there is experimental and calculated terms included in your definition. And that's why it's semi-empirical atom model or method. So now you have got introduced to a semi-empirical interatomic potential. In the next lecture, we will see another type of popular interatomic potential, that is Tersoff potential. Thanks. Hello and welcome to the 6th lecture, Molecular Dynamics Force Fields, where we will discuss on Tersoff force field. So we're continuing our discussion on molecular dynamics theory, and we have already introduced the empirical pair potentials, that is Lennard-Jones potentials, and in the previous lecture we have discussed interatomic potentials, EAM, embedded atom model, that is a semi-empirical model. And currently, we are discussing interatomic potentials, a new force field that works on and prefers bond order to define the potential energy in a system. As a flashback, as you can remember, that embedded atom model thinks as if the atoms are immersed in an ocean of electron density field. So aside from the simplistic definition of the force field or interatomic potential, we have to also include this electron gas density term in our definition. And now Tersoff potential, which is also a very popular potential. It is a bond order potential. Let's define what is bond order. Let's say in a system, when you are defining a molecule, there are several number of bonds, right? Possibly there are several ionic or covalent bonds. The bond order is when you consider two atoms, the number of covalent bonds shared between these two atoms is called their bond order. So this is also you can take into consideration when make a force field. So aside from their position, if you consider their bond order and add a term related to that bond order, then your force field actually becomes more comprehensive and more realistic because you are considering the bond order and covalent bonds are taken into consideration. And when you do that, it's actually Tersoff interatomic potential. So you can see the functional form here. And this is just a map like how the potential energy actually varies with respect to interatomic distance when you actually differ or change the bond order in the system. You can see that you can make a map of interatomic distance versus potential energy in the system. So that's kind of a high-level introduction to Tersoff potential. Right now, it's not necessary to go into the very detail of how these bond order potential works or how pair potential or EAM works, but now you can get a vibe that how a system is defined in terms of potential energy. So in the next lecture, we'll continue our discussion on force fields or interatomic potentials. Thanks. Hello and welcome to the 7th lecture, Molecular Dynamics Force Fields. And we are continuing our theoretical discussions on molecular dynamics theory, especially on the interatomic potentials. So previously we have talked about a very popular potential, that is Tersoff, that is a bond order potential. We have introduced you to empirical or Lennard-Jones potential, semi-empirical or EM potential, and Tersoff, or that is a bond order potential, to show you how differently you can define your system in terms of potential energy. And now we will continue our discussion on the force fields or interatomic potentials. This is just a flashback of the previous Tersoff potential lecture, where we have defined, where we have described how to define potential energy in terms of not only the position, but also the bond order. Now we have an overall idea what is a force field. A force field is the very definition of the system in terms of potential energy, the energy state of the system. And there is a governing equation that is an energy function. That's what we have been showing in terms of functional form. And it is required for any system you want to build. Let's say you want to make a protein system or a biomolecule system or a polymer system. You have to define the governing equation, how they behave and interact with each other. And let's say you want to define the whole system, right? In the simplistic definitions of pair potentials or semi-empirical methods, you can see that we have captured the system in terms of the position of the atoms or electron density or a bond order. So we wanted to capture the behavior of the system. But if you want to define a whole system, you have to actually define several terms to capture the whole system. Let's say if there is a polymer or a protein, there are bonds, there are angles, there are dihedrals, that is angles that are not in the same plane, and also some non-bonded interactions for charge, that is Coulombic interaction, or for position like van der Waals interaction. So if you want to define the whole system realistically, you have to write terms corresponding to these parameters like bonds, angles, dihedrals. And when you do that in combination, it is called a force field. So as an example, a very parameterized model which works well for biomolecules is called CHARM force field, or CHARM potentials. Its potential energy, previously we have seen E, but U, now we are seeing V, but is the same as the potential energy. You can see that there are several terms that are defined to capture the whole system. There is a bond term that actually defines the whole system's bonds in terms of harmonic springs. And then there is an angle term which talks about the angles that are existent between atoms. There are also dihedral terms, improper terms, Urey-Bradley terms. You can search for all of them, and a documentation page from LAMMPS or a CHARM force field will pop up, and you can go through the details of the theoretical description. And there is also non-bonded interaction, as you have mentioned, that there is electrostatic, and there is for positional attraction and repulsion terms, as you have seen. So now we have introduced you to the simplistic versions of pair potentials so that you can understand. But in reality, when you define a force field or interaction potential, these many things you have to take into consideration. So it will be very convenient if you know the names of the popular ones for classical molecular dynamics, where the atomistic level of interactions are taken into consideration. CHARM, amber, OPLS, chromos, these are very popular ones. Now, for example, CHARM is used, CHARM or amber, these are used for biomolecules. There are also sometimes your system is very big and you cannot actually capture all the atomistic interactions. That's why you have to coarse grain them. That is, let's say a set of atoms is considered as one big atom or a seed. This way you can also make a force field. For example, in this way, Martini force field works. Sometimes you want to you want to capture the reactions that is existent in a force field. There's a typo here. It's actually reactive. So in this regard, a reacts FF force field is very popular one. So why so many force fields? Why don't we come into the middle ground and define everything with a single equation? Because there are tons of different behaviors and structures and functionalities of the systems. The way one type of molecule behaves, the other type of molecule behaves does not behave this way. So we have to come up with so many functional forms, and we have to actually fine-tune or tailor the parameters and optimize for a particular type of systems. That's why you will see that sometimes some force fields work for metals, some works for polymers, some works for alloys, and some simplistic ones do not work well for many systems. And sometimes you can see that for biomolecules, there are specific parameterized force fields which do not work for other systems. Sometimes some force fields will work for membrane systems, some will work for protein systems, and that's why you have to actually change the terms or change the functional forms or the parameters or constants associated with them. And as a result, you have to come up with so many force fields. And sometimes you have to also improve over the previous version of a force field. So you have to be very careful which version you're using and which terms they're taking into consideration. So in terms of underlying math, what is going on in a force field, what you are doing is that you are actually solving Newton's equation, as you already know.
and you are constantly performing an integration for the whole system, and their solution, this solution of this integration actually gives you the trajectories of the atoms that is present in a system. So this is kind of a functional form how you discretize a solution in a system, but I'm not going into many details because at this level, high-level introduction is important, and that will give you a sense of ease to understand the material. And also the documentation of the theoretical background is very, very detailed if you search for it, like how a force field is working in terms of solving the underlying equations. But for now, I believe that this is an appropriate level of extent of detail that I'm going through. And as you can see that you are solving these equations for so many times. Let's say you are running a 1 nanosecond of simulation and you are solving this equation to get the atom trajectory so many times. You are solving it every few femtoseconds or every picosecond. So in order to optimize this, in order to calculate this correctly and optimize this for your system, a very big system contains several thousand atoms, there are several ways or algorithms to do it. For example, Verlet or velocity Verlet algorithms, which I'm not going into detail, but it's good to know what to search for when you want to know more detail about the underlying map. So long story short, in order to get the trajectories of atoms, the movements or behaviors or interactions of the atoms, you are actually solving Newton's equation. As we have mentioned in a previous lecture, you can revise it. We have actually shown what goes on in a molecular dynamics simulation. And how often, as we have talked about, that we're discretizing the problem, we're solving the problem, we're solving the equation at a defined interval. How often are we solving that? The answer to these is the definition of the time step. How many times or how fine-tuned you have to do it in terms of solving the equations related to the molecular dynamics. You can see that atomic vibration in lattice, it's actually 10 to the power minus 14 second. That is a few femtoseconds, tens of femtoseconds. And that's why if you want to capture the movement of the atoms or the behavior of the atoms, you have to actually solve them for 10 to the power minus 14 or less. That is 10 to the power minus 15. Let's say you have to solve the equation every femtosecond, 10 to the power minus 15 seconds, in order to capture what is happening at the atomic level in the lattice. So the answer to the question, how often do we solve, is it is generally in line with the atomic vibration, the frequency of atomic vibration in lattice, that is 1 femtosecond generally, in order to capture. If you solve it at a bigger time scale, let's say you are solving at every 1 second, then you are not actually capturing anything, because by the time you have solved it, it has actually vibrated so many times that you will miss the details. So in order to capture the detail of what is happening in the system, you have to solve, you have to set the time step of solving according to the atomic vibration or whatever phenomenon you are trying to capture. So now we have gone through the atomistic detail of the molecular dynamics setting. Now in the next lecture, we'll talk about the molecular dynamics ensembles, that is the holistic definition of a system that goes on in a molecular dynamics simulation. Thanks.
Hello and welcome to the 8th lecture, Molecular Dynamics Ensembles. So we have been discussing the molecular dynamics theory, and in the previous lecture we have introduced you to the concept of force fields. We have been actually engaging in multiple lectures to explain the concept of force fields, and we have explained that in order to define a system, you want to put it in terms of the potential energy of the system. And if you want to express the potential energy of a system in a functional form, then you have to define so many terms. And how you define those terms, it actually depends on your system. And sometimes you define force fields or interatomic potentials by focusing on atom-to-atom interaction. Sometimes you consider more things like the rho, the electron gas density. Sometimes you include more things like bond order. Sometimes in other force fields like term, you include so many terms like bond angles, dihedrals, and non-bonded interactions. So, but when we talk about these, we are actually talking in the very atomic scale. But if you think about a molecular dynamic simulation, it's actually running an entire system, and you want to get the macro level properties by running an atomistic level simulation. And when it comes to macro level properties, you have to actually define an ensemble. Just a flashback that we are actually solving Newton's equation to get the trajectories of atoms, and there are underlying algorithms or ways to solve these kind of equations. And we have also introduced the concept of time step. So as we are talking about ensembles in this lecture, so far we know only about the atoms, the atomistic scale or level of interaction. But in a system, there is not only 2 or 3 atoms that we can talk about. Let's say there's a protein that is solvated in a simulation box in 1,000 of water molecules. So if you think about the holistic system, as I'm showing here in these figures, in this simulation box, there are actually thousands of atoms. So when you consider their macroscopic state, you are actually solving microscopic or atomistic level of equations, but you are trying to get macroscopic state. And that's why you have to use some macroscopic level of conditions on the system. And collectively, these conditions are called ensembles. So what kind of ensembles can we actually apply on a system? There are generally 3 types. One is called microcanonical ensemble, or NVE, in which N, V, and E of the system, that is the number of atoms, the volume of the system, and the total energy of the system is kept constant. Another one is canonical ensemble, where the NV and the temperature of the system is kept constant. And the third one is isothermal isobaric ensemble, where number of atoms, pressure, and temperature are kept constant. You can think about experimental constraints that are actually applied when you perform an experiment in real life. That similar thing you are doing in simulation here at macroscopic state. So that's how ensembles work. It will be more clearer when we will talk about molecular dynamics simulation input script in detail. So in the next lecture, we'll talk about another thing that comes in handy when you run molecular dynamics simulation, that is periodic boundary conditions. Thanks. Hello and welcome to the 9th lecture, periodic boundary conditions. So we have been continuing the discussion on molecular dynamics theory, and previously we have introduced you to the molecular dynamics ensembles. In the current lecture, we'll talk about another important aspect that you apply on a molecular dynamics simulation, that is periodic boundary conditions. Just a flashback, we have introduced you to NVE, NVT, or NPT ensemble that you apply to a molecular dynamics simulation to get its macroscopic state. That is similar to experimental conditions. Now, in order to introduce periodic boundary condition, what we do is that we can imagine a cube. Although I'm showing a 2D image, imagine a cube in which the central cell is actually extended infinitely in all directions, X, Y, Z, where all the cells is actually a copy of the central cell. And
There are infinite images in all directions. Think about a 3D cube in which at the center there is a central box in which the actual simulation is happening, and all the other boxes are copying what is happening at the central box, and there is no wall and no surface molecule. If it happens for an atom to leave the box from one side, it will enter the other side and it will keep the system as it was. So that's how periodic boundary conditions work. And why it is needed? Because we want to know the bulk property. And when it comes to only one cell, it's really small. And if we want to get a bulk property of a system, we have to actually consider an infinitesimally long system that is extended towards every direction. And you will see how it is applied in an input script in a molecular dynamics simulation. So in the next lecture, we'll also introduce another concept of energy minimization. Thanks. Hello and welcome to the 10th lecture, Energy Minimization. We have been continuing our discussion on molecular dynamics theory. And in the previous lecture, we have introduced you at a very high level to periodic boundary conditions, which is needed to run a molecular dynamics simulation in general. And now we'll learn about another concept, energy minimization. So this is a flashback of the previous lecture where we have shown that a central box is actually extended or copied infinitesimally in all directions to get a macroscopic level of properties or a bulk property. Now, let's say you have a structure or a system or a simulation box to run, but if you run the simulation with the default mechanisms without doing any fine tuning, energetically speaking, it will most likely not run because the default coordinates or the conformation of the atoms in the system may not be energetically viable or optimized. Because remember that we are defining the behavior of the system in terms of potential energy. But you have to actually minimize the potential energy of the system before running any simulation so that you do not run into any error, energetically speaking. So that's why you run an energy minimization algorithm on the whole system before running the actual simulation, and that's called energy minimization of the system in general. And if you talk about a very high level definition of it, it is actually altering the conformation of the atoms in the system so that you can actually know another conformation that is actually at a local energy minimum of the system. So the system will try to find out at which conformation the total energy, total potential energy is at a minimum state to begin with. And then from that state, you run the actual simulation. And to get the energy minimization, there are several default algorithms that come into play. For example, steepest descent algorithm or conjugate gradient algorithm. And there are many others for which there are detailed documentation. But as a general scheme, if I want to define it, here, the energy tolerance and the force tolerance are generally set up, and it is run for some iterations to see whether the system can go into very low local or global minima of energy to begin with the system to run the actual MD simulation, and to find whether the energy and displacement of the system is close to zero. It will run for some iterations and try to validate this set of conditions to get to the initial energetically viable condition. And that's how you run an energy minimization of an energy of an MD simulation. And so far I have covered the overall theory of molecular dynamics that will be sufficient at a high level to run a molecular dynamics simulation. And in the next lecture, we'll go through the overall MD review and scope. Thanks. Hello and welcome to the 11th lecture, overall MD review and scope. So we have been discussing about molecular dynamics theory for the previous several lectures, and in the immediate earlier lecture, we have actually discussed about energy minimization, and in this lecture, we will just get a review of MD and its scope. So in energy minimization, we have said that we want to run an MD simulation, but before that, we have to actually minimize the total potential energy of the system by running some algorithms by this general scheme, by setting up some energy tolerance and force tolerance and running it for some iterations to find whether after some iterations the energy and displacement is close to zero, so that we can be sure that the current conformation is energetically viable to run an MD simulation. So now, as I have introduced so many concepts to you in the previous several lectures, now I think that it will be very helpful to get a review of what you have learned so far in terms of molecular dynamics theory and scope. So let me give you a flashback that MD simulation means solving the force F equal to ME equation, and we want to determine the trajectory of the atoms through this atomistic model by solving this equation continuously after several time steps over and over again. And then we analyze the data and output results, whether it be force, displacement, or stress, strain, or something like that. So in terms of scope of MD, what we can and can't do, we can solve problems which includes microseconds of simulation and systems that is nanometers of length. And we use some force fields that are approximated, not parameterized particularly for our system. So we may have to do some tweaking according to our system, and sometimes it's not possible for some particular problems. And it's computationally expensive, especially if you run for several microseconds, or if you run for a very large system. It will cost you hours and a lot of money, because the high performance computational units are generally purchased by your institution. And sometimes your insight that you are getting by running the molecular dynamics simulation, it might not be relevant to your system. It might not be even needed. And sometimes you get partial insight because your problem may need some electrical, optical, or magnetic property insight, but you are not getting it from MD simulation. So MD simulation, long story short, MD simulation is not just the only answer to all the questions according to your research. You have to find out whether MD simulation is the particularly appropriate approach to solve your research problem. But we can definitely solve a lot of mechanical, structural, biochemical, or thermal problems, and we can get particular insights at atomistic level by using this particular method or tool. So that's why we use MD. And if we want to pose it in terms of biomechanical or chemomechanical problem, we can solve these type of research questions, like whether in a simulation, whether a particular protein-to-protein interaction is getting affected, whether we can determine the atomistic level of deformation by using computational mechanics principles, or we can get a lot of atomistic level of insight. But if your system is too big, we'll probably have to leave atomistic approach to solve a problem and go to continuum level of modeling or experimental approach if it's appropriate. So it's very important to really think through your problem to find out the most appropriate solution to solve your problem. So from the next lecture, it will get more interesting, I think, because we will introduce you to the concept of MD software. Like we have learned about how MD simulations theoretically works, what goes on under the hood, but now you will know what software or what packages to use to run a particular MD simulation. So I will see you in the next lecture. Thanks. Hello and welcome to the 12th lecture, Introduction to MD Software. So we have ended the discussion
on molecular dynamic theory in the previous lecture, and we have discussed about overall MD review and scope. Now we will introduce you how to run, introduce you to the concept of how to run molecular dynamic simulations through servers or softwares. So this is just a flashback of what happens in molecular dynamics and what we can and can't do with MD. So in terms of MD software, there are several packages that you can use. The most popular ones are LAMMPS, CHARM, NAMD, GROMACS, Desmond, OpenAM, DL Poly, and there are many more that are popularly used. And these are, and which package is appropriate for you, that actually depends on what problem you're trying to solve. And for example, if you want to solve a problem associated with some protein or biomolecules, probably you are going to be using CHARM, NAMD, or GROMACS. If you are trying to solve a material science problem, you'll probably use DL Poly or LAMMPS. LAMMPS is kind of a common ground for material science or biomolecular simulation because it supports a lot of systems in terms of material science and biomolecules. And when it comes to package, in a package, there are three types of supports, like an interface where you actually run the commands to run in the simulation through a script or command line interaction. And also there are packages of force fields. We have already defined or discussed on force fields, like these are the conditions in terms of functional forms that you have to apply on the system to run a molecular dynamics simulation under some rules and regulations. And when you are running the simulation under the conditions of a force field and you are interacting with an interface, you will come up with some results and you want to analyze it so that you can produce some data. And in order to visualize those data, or in order to visualize your system, you remember that popular in the simulation results look like vibration of atoms or stretching of a protein, something like that. You want to use some visualization package. Sometimes your package will come with visualization or interface. Sometimes it will not. You will have to use some other separate packages. So some popular visualization software are VMD, Oviedo, Avogadro, Vista, Chimera, Pymol, and so on. And also, like the packages, these kind of visualizations are also kind of preferred by particular problems. Sometimes for biomolecular systems, you want to use VMD. Sometimes for material science, you want to use Oviedo or Avogadro. Something like that. So after you start using these softwares or you go through their documentation, you will understand which software is well suited for which. So that's kind of a brief introduction to the names of the software, and we'll go through what happens in those software when you try to run a molecular dynamics simulation. And in the next lecture, we'll particularly talk about the structure, file, and visualization in an MD simulation. Thanks. Hello and welcome to the 13th lecture, MD simulation structure file. So we have started the discussion on MD software and servers, and we'll show you how to set up and visualize MD results. So previously we have introduced you to the MD software. Now we will particularly introduce you to the concept of MD simulation structure file that you will deal with. And we have already told you that in an MD simulation, when you run through a software, there are three types of things that you have to take care about. One is the interface that you are interacting with of a software that might have a GUI, graphical user interface, or a command line interface. And inside that package, there are packages of force fields that you will apply on your system. And at the end, you will visualize your result. So to begin with, you start with a structure file, the structure file of the material or the biomolecular system that you want to simulate. What is in that structure file? There are actually coordinates of the atom. And if it's a crystal, there are probably additional information like crystallographic information.
If it's a protein or a biomolecule, then probably there will be more information, like which portion are helix, which portion are beta sheet, something like that. So a structure file actually defines the structure that you are going to use for your simulation. And in the visualization, what you are looking at, you see some balls like atoms, or you may see the secondary structure of a protein like alpha helix or beta sheet. In the visualization software, you'll see how does it look like, how does the molecular dynamics simulation and the updates of the trajectories when the molecular dynamics simulation proceeds, how does it look like? That information is you are getting from the visualization software. So when it comes to a structure file, the ending of the or the naming of these structural files differs based on what are they related to. For example, in material science, the most used files are with these endings, like these are probably XYZ file or CIF file, crystallographic information file. And if it is biomolecules, proteins, or membranes, you're probably going to start with a PDB file, a protein data bank file that contains the informations, coordinates, and the secondary structure information of the structure. Sometimes it's a platform-specific file. For example, if you are working in LAMMPS package or the LAMMPS platform, you have to convert whatever structure you have to a LAMMPS data file. It doesn't have a definite ending. You have to convert it to a LAMMPS data file, and it has its own syntax and format, and you have to convert it according to that. Now, how do I open or read or visualize these files? There are several options for that. Those are also particular to your system or your approach to solve the problems according to your field. Several popular options are VMD, Chimera, Ovido, Vista. For example, a biomolecular system, generally you use VMD or Chimera. For material science, generally use Ovido or Vista. So long story short, in material science, generally you start with a structure file with XYZ or CIF. And if it's a biomolecule, you start with PDB. If it's a platform specific, you have to convert it to a LAMMPS data file. So how does it look like? Let's look at some examples. For example, I have taken it from the Wikipedia page of XYZ file. If you search for XYZ format, how does it look like? You can see that for the pyridine molecule, the XYZ file, if you open it with a text editor, it will look like this. It will show you how many atoms are there. You can see that there are 11 carbon, nitrogen, and hydrogen atoms, and you will see their coordinates in the 3D space, the X, Y, Z, and coordinates of the 11 atoms. So that's how an XYZ format looks like. It's really simple to interpret. If it comes to a PDB file, let's say for a protein, it's actually an extract from a protein PDB file. You can see that it will show so many information. For example, it will show the indexes of the atom. You can see that there are 12 atoms here. It will show there are nitrogen, carbon, oxygen, and what kind of carbons are there, and what kind of nitrogen are there, and what residue it belongs to. For example, lysine, for example, valine, and which chain it belongs to, and which residue it belongs to. You can see that the 11111, that is actually the lysine residue, and here the valine is beginning, and the actual PDB file is actually much more, which cannot be accommodated in this single slide. And you can see there are coordinates as well, which atoms are there, as well as there are other additional information. So when you open a PDB file with a text editor, that's what you look at. You can get so many information about this structure that you are going to simulate. And if it comes to a LAMMPS data file, it also has similar type of information. For example, it will start with how many atoms, bonds, and angles are there, how many types of these are there, and what is the box size in terms of X, Y, and Z coordinates. It will show you the box size in terms of angstrom or whatever unit you have used. What masses are there? You can see that there are 16 and 1, that is oxygen and hydrogen, are there. There will be some molecular dynamics pair coefficients and bond coefficients and angle coefficients are there. Then there will be atom information. For example, their indexes, their coordinates, and so many more information. So that's how actually a LAMMPS data file looks like. This is actually a structure file that gives you an information of all the atoms contained in that system, in the structure. So where do you get this structure file? Sometimes you have the ability to create it from the scratch inside the platform. For example, in LAMMPS, you can create the, in LAMMPS or VMD, whether you are working on a material science project or a biomolecular project, you can start it from the scratch. Sometimes it's not possible. Sometimes you have to go through some database. For example, popular material science databases are Material Science Project, Open Material Science Database, crystallographic database, or specific research groups that make their particularly useful structure files and deposit in their repositories that can be accessed publicly or sometimes upon request. And if it comes to biomolecules, RCSB is possibly one of the biggest databases for structure files, biomolecular structure files. Sometimes you get the structure files from a UniProt or PubChem, and again, from specific research groups, repositories. So the structure file sources can be diverse and multifarious, and you have to determine how you want to make your system or how you want to start with your structure. So these are some homepage screenshots for these databases that I have told you about. For example, this is a material science project, which is very popular for material science related projects. You can get a lot of structures from there. Probably you have to sign in or register to download the structures in XYZ or CIF format. This is open materials database. You have to search for a particular alloy or an element, and when you submit your search, you will see how many structures associated with your search are available in their database, something like that. This is crystallographic open database where a lot of crystallographic information and structures are deposited. And this is a very popular one, RCSB, where protein and protein-protein interaction structures are deposited. For example, you can see here is a structure of hen egg white lysozyme that is a protein, and you can download this file in a PDB format or FASTA sequence, the amino acid sequence from here. And also this is UniProt. Here you can search for it. For example, this is also lysozyme. Chick means it's for hen lysozyme that I have shown you here. And you can also download in a format from here. And also there are a lot of additional information when you search for a particular protein or molecule here, as well as any other database. Feel free to explore through them, because a lot of information can be achieved from there. For example, how this structure is obtained from X-ray diffraction, and there will be the publication associated with it so that you can learn from it. And this is a very popular chemistry related database, that is PubChem. You can get a lot of chemical structure from here. Aspirin, sometimes you have to download the structure and convert it to another format. For this kind of conversions, you also have other software or facilities like OpenBabel or so on. And you can see that there are so many other functionalities and tons of information associated with a particular molecule. And this is an example of a structural file source from a specific research group. For example, this is J.B.K. Loda lab of molecular and thermodynamics modeling that actually has deposited so many lipid membrane coordinates that they have worked on, and you can download it from there in different formats like PDB, CRD, PSF. So that's how we can get your structure files or make your structure files. And in the next lecture, we will talk about visualization of the simulation.
Hello and welcome to the 14th lecture, Md Simulation Visualization. And we have been continuing our discussion on setting up and visualization of Md in terms of software and server. And in the previous lecture, we have talked about the structure file, on how to attain them or make them. And now we will talk about how to visualize our data. We have talked about the different formats like XYZ, CIF, or PDB or LAMMPS data file, and how to use those files through some software. And we have shown you some examples of databases and research groups. So when you want to visualize the Md simulation results, very popular platforms of software are Ovido and VMD. Also, there are several ones, Chimera, Pymol, Vesta, and so on. So as an example, if you open a data file or a data dump file after an Md simulation is run through Ovido, then you see something like this. For example, there are so many molecules packed in here and color-coded according to their elements. And you can do several modifications through here. Like here you can see that it's called data.h2o equilibrium. It's a LAMMPS data file, and it's possibly a bunch of water molecules packed together in a simulation box. So probably it's just a water box. And you can see the information in terms of simulation cell bond types or particle types, and you can add some modifications in here, like select if you want to select some molecules or delete some molecules, or if you want to just see the simulation, then you have the option. Another popular one is VMD. When you open VMD, there is a visualization window like this, which will show you the system or the protein that you are simulating, and it will show you some information like number of atoms and frames, and you will have so many options to modify or manipulate your system or make a good graphics animation video. Or you will have, through the extensions, there will be a ton of options to analyze your data. So these are some examples on how to visualize the data. The usefulness of these software or platforms that actually come through use. So I'm not going into too much detail of how to use the software, because a software cannot be learned through PowerPoint presentations. You actually have to go through clicking through the buttons of the software. So I have given you a very high-level introduction to the software and visualization. Now let's go through the next lecture in the simulation potential parameters. Hello and welcome to the 15th lecture, Md potential parameters. So far, we have been discussing setting up and visualization of Md. Previously, we have shown you some software regarding Md simulation visualization. And in this lecture, we will talk about Md simulation potential parameters. These are some examples from Ovido and VMD, like how to visualize your system. And as of now, you should have an idea of Md simulation. It has three critical elements. One is a structure file that we have discussed. One is the visualization, that you will visualize the system by using which approaches. Now, we have also told you that in the software platform or package, there are packages of potential parameters that we have already discussed. Now, where do you get those potential parameters? The answer is whether you can build those parameters or you use some already determined parameters, a set of parameters in your system. So it is really easy to use some already pre-built set of parameters in your system if it is available for your particular scenario. If it is not, you may have to build it on your own according to the literature. Sometimes these potential parameters are available from the databases, for example, through IPR or WebFF, some popular repositories. Sometimes, just like structures, you can obtain them through specific research groups, which particularly engage in developing potential parameters for particular systems. For examples, if you go through this database, IPR,
then you can find that there are several pre-built potential parameter sets for alloys, compounds, and also fictional potentials that have not been validated by experiments, but they have developed them according to the theoretical ground. So there are tons of resources in terms of potential parameters, but which one to use here? The answer is it depends. You have to go through the literature review. Like, let's say you want to simulate system A, there is not any resource or literature on system A, then you have to go through the related systems that are particularly associated with system A, and you have to get an idea on what parameters are actually relevant to this system or might be realistic to use on your system. So you have to really consider experimental parameters. Sometimes you have to tweak some parameters of some pre-built potentials according to your particular problem. And you have to keep in mind that you just cannot plug and play in terms of using potential parameters, because a single potential is not good for everything. If you go through the previous lectures on potential parameters, you can get an idea that sometimes in a potential parameter set, only atom-to-atom interaction are taken into consideration. Sometimes electron density is taken into consideration. Sometimes bonded and non-bonded interactions are taken into consideration. Sometimes reactions are taken into consideration. But a single potential is not parameterized for all the systems. Some, even for biomolecular systems, some systems were good for proteins, some work well for membrane systems. But a single system, a single force field that is not good or well parameterized for every system. So that's why you have to think really critically about selecting your force field parameters when you are trying to run a simulation. So in the next lecture, we'll show you how to actually run an MD simulation in LAMMPS. Hello and welcome to the 16th lecture, running MD simulation in LAMMPS. So we have been talking about so many things of MD simulation, like how to understand the underlying theory, or how to use a software associated with molecular dynamics simulation, how to visualize, how to get the structure file, or how to get the potential parameters. Now we will introduce you to an MD platform, LAMMPS, that is kind of a common ground for running molecular dynamics simulation because it supports so many systems associated with material science and biomolecules. So we have already talked about how to use the databases and and how to choose between the databases, or how to make your potential parameters. Now, when it comes to running MD simulation in LAMMPS, we need three things to run MD simulation. One is the structure file or data that you will obtain or you will build. You will need potential file or data that you will obtain or you will build, or sometimes it's integrated into your package. And you will need an input file that will contain a bunch of commands to tell the system how you want to simulate the system, or what you want to do with your structure. And it will also contain commands related to visualization, how you want to visualize or what data you want to extract. So these kind of things, you will submit it into a package. The package might be LAMMPS, it might be GROMACS, it might be NAMD and CHARM. For our case, let's say that we're running our system in LAMMPS. So we will run our simulation in a package in LAMMPS, and for that we can do it in two ways. We can make our system from the scratch through our input script. In our input script, we will tell the system how I want to make my simulation box, what conditions I want to apply on it. So for that, you have really good tutorials available online, like Evolucity tutorial LAMMPS. It works on LAMMPS. And it will be really helpful if you're from material science background. Or if you want to see some tutorials on biomolecules, there is Dr. Justin Lemko's tutorial that works on with VMD. Sometimes you can also make the system from a server itself. Let's say you don't want to make the system from the scratch with your input script, rather you want everything to be made beforehand, and if you want to just modify a few things and run the simulation, that option is also available. For example, a popular server is Charm GUI that actually facilitate you to submit just a PDB file and make the whole system. It will solve for you, it will add the ions for you, and it will make or convert the system to LAMMPS readable data files for you so that you can just run the equilibration or production simulation. So in the next lecture, we will show you the first option, like how to run an MD simulation from the scratch, and it will I will walk you through briefly from a tutorial, LAMMPS Evocity tutorial. Hello and welcome to the 17th lecture, running MD simulation from the scratch. So we have been talking about the MD platform LAMMPS, and previously we have given you a high-level introduction on how to run MD simulation in LAMMPS, what's in there. But now, and we have talked about two ways of running the simulation. One is by making the system from the scratch, and another one is making the system from a server. So in this lecture, we'll talk about the first option, that is running MD simulation from the scratch. And to facilitate these, we'll go through a very popular basic level introductory tutorial in LAMMPS, that is Evocity tutorial, which calculates the cohesive energy and lattice parameter for aluminum. And for that, let's go to Google and search for Evocity LAMMPS tutorials, go to the first one, and you can see that it shows how to go through from the beginning to the end of a simulation setup. And here it comes. We have told you that you need three things to run a simulation. One is a structure file, one is potential parameter, and one is the visualization. So for that, everything is facilitated here, and here is the example input script to run this simulation. Let's say you want to know the lattice parameter and cohesive energy in aluminum, and for that you want to make the system from the scratch. So there are several parts of an input script that you will submit to the system. For example, here is some comment lines that explains what is going on here. There are several sections that are denoted by the comment lines. For example, you are initializing the simulations and you are telling the system how you want to define the system in terms of dimensions. It has 3 dimensions. It has 3 periodic boundary conditions, and what atom style and atom modify for this. So if these are not relevant for you, or you find difficulty to understand these, always remember that there are tons of documentations that you can go through to actually understand this. For example, for all these keywords, units, dimensions, there is a specific separate documentation page of LAMMPS. For example, let's say you don't know what units metal means. So you just search for it. LAMMPS units. You can get the first search and you will see that it's a LAMMPS documentation page, which actually explains the units command. For example, how to write it, the syntax, what is the style of it, and what does the keyword means. For example, if you're using style metal, then the units will be this. Your mass will be grams per mole, your distance will be in angstroms. So throughout your system, you are telling your system which units to use. And also you can search for what dimension means, what boundary means, what might be the other boundary conditions if you don't want to use PPP. And then you create the atoms. That means that you are creating the systems, the simulation box. The lattice, you are telling that it will be FCC. You are defining the region, you're creating the box. And then you define the interatomic potential. It has actually given you some instructions on how to get the potentials. For example, interatomic potentials, click here. For aluminum, click here and click on whichever potential you like.
they have also given some download link for the potential files that you will that you will download and paste it in your folder along with your input script in a text file. And then they will call these files. That is, they are calling these potential parameters to use in your system, so that according to that potential parameter, your system is actually made. Then you are defining some other settings, compute ENG, compute E-atoms. That is, you want to compute some energy and compute how you want to compute the energy of the atoms. Or it might be something else. You can search for compute ENG or compute E-atoms, and you will end up with reading a documentation page of LAMMPS. That's how you will learn in the fly. I'm not going through the technical details because you will have a very detailed description of all the lines. Now you will run the minimization. As we have told you that before running the system, we want to minimize the potential energy of the system. And it is applying some fixes on the system. It is telling how frequently you want to document the thermodynamic properties of the system and in which style. And then it is running the minimization. I have told you that it is setting up some energy parameter, energy tolerance and iterations. And then it is defining some variables that will come in handy to make your calculations. That at the end, it will print your calculated values, like total energy, number of atoms, lattice constant, and cohesive energy. And at the end, it will print all done. So if you have LAMMPS installed in your system, then you can run this simulation through this input script. And there are several methods on how to run this simulation. It has described it in step by step, and also in so many platforms like Windows or Unix, and what it does. And it is actually going through step by step on how to interpret your results. So that's how we can make a system from the scratch and run an MD simulation. Don't worry if it doesn't make sense because you are not from the same background. It's actually a very common simulation. If you are not from material science, if you're more interested in biomolecules, it will still come in handy if you want to use LAMMPS for your simulations. So let's go back to our presentation that we have been talking about the first method of running the simulation from the scratch. In this next lecture, we will talk about how to run an MD simulation from a server or a software.
Hello and welcome to the 18th lecture, running MD simulation from a server or software. So we have been talking about introduction to MD platform LAMMPS, and previously we have shown you how to run an MD simulation from the scratch. And now I'll show you how to run an MD simulation from a server or a software. As a flashback, we have shown you an Evocity tutorial where we have shown how to calculate the cohesive energy and lattice parameters for aluminum. And now we'll show you a server called CharmGUI, which is actually an input generator. After you create an account in this server, you will find this many modules and functionalities where you can create your system. It is an input generator, meaning that if you submit, let's say you are in the solution builder module, you will get an introduction on the steps, what happens here. And you just upload your structure file, that is PDB file, and then you go to the next step. And in every step, you will see what happens. First, it will read your coordinates, that is the structure file. It will solve it, the molecule. It will add the ions. It will set up the periodic boundary conditions, and then generate the input files required to run the simulation, that is equilibration and production files. So if you don't want to create your system from the scratch, that is, if you don't want to write a very large input script, rather you want this portal or server to create all the required files to run your simulation, then you can use this CharmGUI server to run your simulation. If you're still confused, then you can watch these 3 videos to get a more detailed and segmented ideas about running an MD simulation that you can find in this playlist, MD simulation. How to install LAMMPS in Windows 10 PC and run an MD simulation, and LAMMPS tutorial, where I have shown step-by-step installation, creating the system, running simulation, and how to fix the errors that you can encounter in an MD simulation, and particularly how you can use OVITO to visualize your simulation data and how to interpret them. So in combination, if you watch this lecture along with these 3 lectures, then probably then I can hope that you will have a more detailed idea on how to run MD simulation by using software or servers. In the next lecture, we'll talk about how to analyze the data in MD simulation. Thanks. Hello and welcome to the 19th lecture, MD simulation data analysis. So we have introduced to the MD platform LAMMPS, and previously we have shown you the second approach to run an MD simulation, that is from a server or a software, instead of making the system from the scratch. Now we'll give you a brief review on how to analyze the data that you get from running an MD simulation. So as you already know from the previous lecture, that we can run an MD simulation from Charm GUI software that generates all the required files to run an MD simulation. And to get a vibe of MD simulation data analysis, we will go through the Evocity tutorial, where we will deform a 3-dimensional periodic simulation cell in uniaxial tension for aluminum. So let's go to the tutorial file, LAMMPS Evocity tutorials and 3. So you can see that how it looks like the tensor loading of an aluminum single crystal movie, and the workflow is kind of the same as we have seen in tutorial 1. You start with the input script and you initialize by defining the system in terms of units and dimensions. And then you create a box by defining the lattice region and associated parameters. And then you apply the force field that you can download according to the instruction on this page. And then you give some setting and then equilibrate it. And you start with some random velocity for the atoms, and then you apply an NPT ensemble on the system and temperature 300. And then you run an NPT equilibration, and you give the system some commands in terms of how frequently you want to see the thermodynamic data and which data you want to see, and then you run it, and you store the data in terms of some variables, and then you calculate the deformation in terms of applying some fixes, that is applying some more commands in terms of strain rate, and how it shows how to show you the strain, how to calculate the stress and strain, and how to store them. And at the output, you will find the stress and strain printed in a file, that is saved in a file, and you will also dump all the data in this dump tensile.cfg configuration file that can be analyzed by AtomEye software. And then you display these thermodynamic data, and you run this simulation, and at the end it will print all done if run successfully. And it will go through step by step on how to run the simulation, how to understand all the sections, and how to interpret the log file. A log file is a file that stores everything that happened throughout the simulation. So at the end, you will get the list of what you commanded the system to store. And it also shows how to plot the stress and strain through a MATLAB script so that you can see a really nice plot of stress versus strain. So generally, in a mechanical tensile loading experiment, you observe stress versus strain data. In other related experiments, probably you want to see force versus displacement curve. If it is a biomolecular simulation, you may want to see other biomolecular characteristics, interaction, and something like that. And it also shows you how to make a movie from it so that you can actually visualize the deformation. So that's kind of a very high level introduction on what kind of data we can analyze through MD simulation. And another example is to for you to try yourself in the second approach that we have shown you how to run a molecular dynamic simulation from Charm GUI. You can actually try to create a system. My suggestion is to explore all the modules, especially solution builder. Use just any protein PDB file and go through the steps of the solution builder to make your system. And at the end, make some output file, input file generated for LAMMPS or NAMD, whichever you prefer. And you can also use membrane builder if you are really interested in solving membrane-related research questions to see how you can create membrane system through Charm GUI. So that's kind of a take-home task for you that will really help you to play with the software and explore it and learn from it. So that kind of pretty much summarizes the software aspect of this lecture. And in this next lecture will be the final lecture of this course, that is MD simulation resources. And I will conclude you by giving you some good resources and how to perform MD simulation practice in real world. Thank you. Hello and welcome to the 20th and the final lecture of this series, that is MD simulation resources and conclusion. So if you have followed this course throughout, and if you have gone through all the lectures, so I would like to thank you and congratulate you, because you have learned about so many things: molecular docking software and servers, a little bit detailed discussion on molecular dynamics theory. You have learned about so many software about MD, how to set up and visualize MD, and how to particularly run MD simulation through LAMMPS, and how to create your system from scratch or from server, and how to analyze the data in LAMMPS. So in this lecture, I will give you a kind of a formal conclusion to this series, and I will give you some information about good resources on MD simulation. So previously, we have shown you an EvoCity tutorial on how to how to perform an uniaxial tension on aluminum. And when it comes to MD simulation resources, where do I learn from? A lot of the times, students are stuck about thinking about which should be my square one to learn conveniently and step-by-step tutorials. So you can use these two resources. One is the EvoCity tutorial, and another one is Justin Lemku's tutorials. EvoCity is generally used for material science related projects, and Justin Lemku's tutorial shows so many biomolecular projects step by step, and he uses VMD and possibly GROMACS. And I cannot emphasize this more, that LAMMPS and GROMACS documentation are very detailed, and you can virtually find explanation of everything if you just search for it. Let's say you search for LAMMPS units. If you come up with an input script for someone else and you don't know what this input script means, my suggestion is to go through every line and search for every line. Let's say units, you search for LAMMPS units, you will find a very detailed explanation of what units means and how to and go through all the other commands to and after you practice with it and apply this to real world projects, you will get a very good idea. And so goes for GROMACS or VMD documentation, Dr. Justin Lemku's documentation, or some software documentation also, like for VMD. They have very detailed documentation and tutorials on how to perform MD simulation on biomolecular systems. Now, generally, the tutorials are made for small systems so that you can learn from them and run them actually in your local PC. But these are actually true for only small systems containing just a small protein or just a few thousand atoms, which can be run in your local server or PC. Generally, computation-intensive labs are facilitated with university cluster computers or supercomputers. They have access to these HPC, that is high performance computational units, that they have to pay for and purchase so that they can run large set of simulations, which facilitates parallel computation and massive computational power. If not, sometimes there are other options like buying these computational packages from third party like Google or Amazon. So now you know how to run these simulations, how to run these simulations at large scale in university setting or lab setting, and what to do now. You have a very technical and high-level introduction on Md simulation. Now your task is to practice and apply this in real-world projects. And if you get stuck, feel free to ask any expert, and also feel free to go through documentation. My suggestion is to go through errors, go through forums, and go through discussions. You may wonder that you are possibly facing a particular problem first in the world, but if you search for forums, you will see that possibly your answer is given 10 to 12 years ago in the molecular dynamics simulation forums. So my suggestion will be to really go through the fundamentals, understand the theories, and apply those in real-world projects. And when get stuck, go through the forums and the documentations of the software so that you can overcome your problems. And always maintain good practice in Md in terms of research. Although it is totally computational, but all the ethics and regulations and other research aspects are applicable for Md simulation, as it is applicable for experimental approaches. So good luck for you, and thank you. Please feel free to like, comment, share, and subscribe.